For most of this class, we work with bacteria, and that's what so far you have been working with. In this exercise, we finally talk about fungi, and we'll work with fungi. Some characteristics about fungi: they are eukaryotic organisms. So that means, of course, they do have a nucleus. The DNA is going to be in the nucleus. Fungi do have a cell wall. It is different than the cell wall of a plant cell, and it's also different than the cell wall of the bacteria. It's composed mostly of chitin. Now, one thing with fungi is that oftentimes people hear of mold and yeast. Mold and yeast are both types of fungi. Differences between them is that mold is multicellular. It grows in typically very long filaments, and these individual long filaments we call hyphae. When you have a whole mass of the hyphae, then it's referred to as mycelium. Yeast are single cell. They're unicellular. Now, some fungi, and it just depends on the different species that you're talking about, some exist only as mold, some exist only as yeast. Some will exist in both forms, both that single cell yeast form and also in the multicellular form. Fungi tend to prefer a nice warm environment. They like it to be moist, and they like the pH to be slightly acidic. And this is one thing we use in our favor in the lab when trying to grow fungi, especially if you're trying to grow a sample that you have retrieved from the environment, so it's not a pure culture, and you may have bacteria in there as well. If you're wanting to grow fungi, drop the pH of the media a bit to make it more acidic. They often prefer pH like 5.5 to 6.5. Remember, pH 7 is neutral. So anything less than 7 is an acid. So they prefer that more acidic environment. Bacteria don't like that. Bacteria prefer more neutral to just slightly basic, like 7 to even sometimes up to 8. So if you want to grow bacteria, bump the pH up a little bit, 7 to 8, somewhere around there. And the fungi aren't going to like that. So you're not going to have, you may still have a little bit of fungal contamination possibly, but not nearly as much as if the pH were lower. If you're trying to grow fungi, like I say, drop the pH down to maybe 6, maybe, and the bacteria aren't going to be happy, so they're not going to grow as well, but the fungi will love it. So how do you grow the fungi in a lab? There's different ways of doing this. If you are growing them on a solid media, typically you would grow them on a petri dish, not in a test tube. What you do is from your original stock plate, you would take a little plug, and then you transfer that plug to a new plate, and then the fungi will grow from that. Now, some people in some labs, for teaching labs, when they're showing you how to do this, to remove a plug, they may actually use like a straw to stab into your first plate that's already inoculated to remove, and that's how they grab a plug and then transfer that and then push the plug out onto the new plate. There is actually this instrument that you can use. I've used it in research where it's a cork bore, and you have you can pull a plug, and you can get them in different dimensions, different diameters, from small to very large, but that way you get a consistent size. And so you can say you pulled a plug of X size when reporting, so that it's consistent, say when doing research. If you're working with a liquid medium, if you remember, bacteria, when they grow in a liquid medium, like in a nutrient broth, they will turn it turbid or cloudy. Fungi don't do that. Instead, when fungi grow in the liquid broth, the liquid's going to remain clear, but they grow in little tiny balls that will be suspended in the media. So if you want to transfer something, you're going to want to make sure you transfer some of those balls. As I said, it kind of looks like the little snow particles in a snow globe. Sabrodextrose agar is specifically an agar that is designed for cultivating and growing fungi. the pH excuse me, is slightly acidic to favor the growth of those fungi. Some people say, well, I thought mushrooms are fungi. Yes, they are. They are the fruiting bodies of fungi. Not all fungi are going to produce the mushrooms. Those that do, there's a definite life cycle and a whole procedure. And you may have noticed this in your yard, that when a mushroom first appears, it looks like the top of it's very closed. The top, or what we call the cap of the mushroom, that's where the spores are. And so once that opens up, it can release the spores. Whether you're working with the mushroom fruiting body, which is often hard to grow in the lab, but even just a regular, like on a petri dish growing fungi, one thing to be aware of is that fungi do produce spores. And so when you, say, open a petri dish, anytime you're working with them, you want to be aware of those spores and not allow them to become airborne and therefore possibly contaminate other things in the lab. So you just have to take some extra precautions with it. There are several pictures on here. This slide up here is showing yeast cells. So they are single cell. You have lots of them in here. They look just like little circles or little ovals. Over here is fungi growing on a petri dish. And you can see it kind of has that classic kind of looking fuzzy on it. You definitely don't get individual colonies like you do for bacteria. This sample over here was actually taken from, we applied a piece of blue cheese onto the Sabarog agar, and then it just allowed it to grow at room temperature. This down here is a picture of a mushroom, the fruiting body, as I said. This top portion is known as the cap. This is the stem underneath it that's holding the cap up. Initially, the cap is very closed, and then as it progresses through the life cycle, it will eventually start to spread out. And now you can see this cap is completely now horizontal. Underneath here where I'm pointing, you can see those are known as gills, and in between those, that's where all the spores are being produced. So by the cap opening up and kind of like spreading up and stretching out, exposing those gills, that allows for easier release of the spores.